In this video, we're going to be learning about how chloroplasts use their specialized structures as well as specialized chemicals that are called pigments to absorb the energy from sunlight that strikes the surface of a plant leaf. Now, from the previous video, you'll recognize these as being grana, which are stacks of individual thylakoids, the operational unit in light harvesting in chloroplasts. And in this video, we're going to be taking a look at the thylakoid membranes specifically, because it's in the membranes of each individual thylakoid in which the light harvesting pigments are found, the most important family of which we can see two examples here, are what we call the chlorophyll pigments, which explain why plant leaves are green for the overwhelming majority of the year. These are the pigments that reflect green light off of the surface of a leaf and therefore absorb pretty much all of the other colors of light. Now, the reason why plants actually make two varieties of chlorophyll, at least, it's actually a very large family of pigments, but the reason why plants use these particular chlorophylls is that each of them, what we call chlorophyll A and B, are specialized for absorbing different wavelengths of light depending on the variances in their chemical structure. For example, chlorophyll A is more specialized at absorbing light in the red, orange, and violet wavelengths, while chlorophyll B is specialized in absorbing light in the blue wavelengths. So it's pigments like chlorophyll, but of course not limited to chlorophyll, whose responsibility it is to capture energy from the particles of light that we call photons that strike the surface of a leaf. Now, without getting too much into detail about the physics of light, photons, in a sense, are a hypothetical particle that explain the dual behavior of light. Sometimes light behaves more like a form of pure energy or pure radiation when it travels in waves, but at other times it behaves more like an elementary particle like an electron, and in which case we give this the term photon, referring to the individual units of light that strike the surface of chlorophyll and trigger the chemical changes that we're now going to explore. So the center of a thylakoid, or at least the central part of a thylakoid in triggering the reaction that is necessary to drive photosynthesis, is a large multi-pigment, multi-protein complex, roughly analogous to a mitochondrial membrane complex. And these multi-protein, multi-pigment complexes are what we call photosystems. And chloroplasts generally have two distinct photosystems, which we call photosystems 1 and 2. The irony, of course, is that photosystem 2 is regarded as the starting point of the chemical reactions in photosynthesis, but we call it photosystem 2, or simply PS2, because it was discovered after photosystem 1. So generally, here is how a photosystem functions in a very simplified form. As mentioned, the primary purpose of a photosystem is to actually do the absorbing of light. So there is a specific chlorophyll molecule that will act as the beginning of the process of light absorption. So when photons of light strike this chlorophyll particle here, it will cause chemical reactions within this chlorophyll particle that will cause it to lose electrons. In other words, we can think of what happens in a photosystem as being a very miniaturized electron transport chain, similar to the one that occurs on the mitochondrial membrane, in that we see two phenomena occur. Number one, the energy from the light absorbed by the first chlorophyll in the photosystem is transferred to the next chlorophyll in the photosystem, and eventually these chlorophyll molecules are going to form a chain like a relay race that cycle the energy absorbed in the form of an excited electron from one chlorophyll to the next, to the next, to the next, until they reach their ultimate destination. And the reason why we can think of this as a miniaturized electron transport chain is because each chlorophyll pigment in the chain wants the energy that is being transferred more than the other one because of increasing electronegativity, and also increasing reduction potential. Each of these chlorophylls gains electrons slightly more easily than the one that came before it. And eventually these electrons are actually going to be transferred out of the photosystem, where they actually enter a true electron transport chain, which is the subject of the next video. What we want to focus on here is what happens at this chlorophyll pigment right here, which is what we call the reaction center chlorophyll. We call this the reaction center chlorophyll because this drives the first chemical reaction that occurs in photosynthesis, where the energy harvested from these light photons that struck the photosystem is actually used to split a water molecule into H+ ions and oxygen gas. Now, this oxygen gas is indeed the same oxygen gas that is released into the Earth's atmosphere and is the foundational basis behind every oxygen-breathing organism being able to survive, all because of this one reaction here. And this reaction has a rather cool name. We call this reaction photolysis. And if we do a word form derivation here, photo is of course the Greek prefix referring to light, and lysis refers to the process of splitting or breaking something. So in photolysis, we are literally using the energy harvested from light to break apart water into two component parts. Now, the other thing that we are doing here is that when the H+ ions are broken off of water and when oxygen gas is formed, we are also liberating a larger number of electrons, and those electrons in turn are funneled into a true electron transport chain in the thylakoid membrane that is used to drive all of the subsequent reactions of photosynthesis. But what we want to focus on is right here. So as a consequence of the reaction center chlorophyll driving the process of photolysis, overall the concentration of acid ions of H+ or H3O+ ions, if you prefer, is naturally going to increase. The more light we harvest, the more photolysis is powered by this reaction, the more water is broken apart, and the higher the concentration of acid is going to be as a consequence of this, meaning that not only are we creating a concentration gradient, but we are also creating an electrostatic gradient because of all the repulsion of each of these H+ or H3O+ ions, and therefore these ions do not want to stay together, and we are creating the conditions that are necessary for proton motive force, exactly as in the mitochondrial electron transport chain. In the next video, we're actually going to be looking at what the harvested electrons and H+ gradient we have created are actually used for, and we're going to be exploring the electron transport chain that is specific to thylakoids and the processes that it is used to drive and how those are used in future processes in the photosynthesis process.